Hello everyone, so I'm Peter Wing, I'm a group leader at the University of Oxford, and it gives me enormous pleasure to introduce Professor Paul Duprex from the University of Pittsburgh, who is going to give the last keynote of this conference about jumping, all about jumping strands from negative to positive. So thank you, Paul, for coming, and we're really looking forward to your talk.
Julian gave me a really hard title, and he's given me a hard job, so that's why I decided I would show you an old picture. And what Julian does really well, and what this group has taught me, is what a wonderful network you folks have built here. I just was walking to dinner last evening, and I walked past a lot of the early career researchers sitting at those tables outside that pub, interacting with each other. And this is really important, right? Because you find your peers in this place. And in the same way as 20 years ago, 25 years ago, Jules and I, Paul Degar, Nick Rose, and many others in this room found each other in meetings like this. So Julian has brought people together. This is San Francisco. We ran the bridge one morning early on. This is Christian, who might still be here. There he is. This is our bike tour around the previous Berlin Wall. And this is the oldest photo here. Where is he? Volker. This is on the Jans Causeway in Northern Ireland many, many years ago. So it's just wonderful to be brought together and talk about jumping strands. So what do we mean by jumping strands? Well, we have to think about the pathogens. I'm in the Center for Vaccine Research in the University of Pittsburgh, and we have an interest in all of these viruses in red we can work with. We have a select agent facility. We do a lot of animal pathogenesis studies. But today I'm going to focus on the first strand, negative measles, the second strand, positive COVID, and then I'm going to jump back strands through both and talk a little bit about mumps so that you know what we've been doing, talk a little bit about what we have done. So here's where we're going in this talk. We're going to talk about the most infectious virus on Earth, talk about the vaccines, talk about disease, and then talk about persistence. That gets me to the positive work that we did, because persistence has been something that has really interested me for a year. So we'll talk about that with respect to COVID, and that follows, I think it was Isabel's wonderful talk. Is it Isabel? Did I get your name right? Talking about the recurrent deletions yesterday. And then we'll talk about things into things. This is doffing my cap to Wendy Barclay, who uses that to describe vectored vaccines. And then finally, we'll talk a little bit about what we're doing with pandemic preparedness. So that's where we're going. Measles. Measles is the most transmissible human virus on Earth. If you remember nothing, remember that. Why does that matter? It's a true airborne virus. It's not transmitted in droplets. It is a true airborne virus, and it's R0. Somebody was talking about R0 earlier. Is anything between 15 and 18. That did not stop Tom Peebles, John Enders, bringing this pathogen into the laboratory to isolate it, adapt it, and then attenuate it at a time when about 8 million people were dying of measles in each year in the world. Now, as the most infectious human virus on Earth, if you don't have anybody vaccinated, of course, everybody gets it pretty quickly. You need to vaccinate because the virus creates lifelong immunity. You need to vaccinate around 95% of the individuals to block endemic transmission. And we in the States did that in the year 2000. We blocked endemic transmission. The virus was eliminated from the US. But nevertheless, here's what we have, 2024, 2025. You can see these this year alone, halfway through the year, we already have 1,300 cases, predominantly in Texas. This is the weekly case report. You can see this big spike. This is the yearly spike. You can see that these already this year, we've surpassed the bigger outbreak, 2020 through 19, that was. So there's currently a big outbreak. We already have 3 individuals dead in the country. We know that that's the case because as soon as you hit about 1,000 individuals infected with measles, the complications, hospitalizations occur, and young children die of this vaccine preventable disease. So this damn thing is confusing me. And why is that? It's because vaccination rates are dropping, and this is the lowest one that I found over in Idaho here. But you can see right across the country, we have a real decrease in vaccine uptake. As soon as you get lower than that 95%, you basically get these little pockets of individuals and the virus spreads really quite quickly. So that's what's going on in the US. But what we need to remember is what happens there often happens here. And now we're in Liverpool and we must talk about this. The tragedy of a 72%, I think is the number I got. You could correct me if I'm wrong in that, but a 72% vaccination rate in certain places is that you end up with exactly the same situation. Outbreaks, local outbreaks, and a child who dies due to measles. So I want to remind you of this negative virus because it's an important virus that we all talk about. We talked in our last session about trusted messengers, why vaccines matter in particular communities. These are the sorts of things we as trusted messengers should be taking elsewhere. Okay. Measles is the most infectious human pathogen on Earth. It's also a really big systemic disease. But way back when I was playing with molecular virology, we made one of the very first recombinant viruses, which allowed us to track, using GFP, the virus in cells. We did a lot of work in cells, but I'm going to show you some slides. Some people have seen this in this room, but I think the younger ones should be aware of what this disease does. So this is looking at the tummy of an infected macaque, which was infected with a fluorescent virus which expresses fluorescent protein. We illuminate it with blue light. We use the filters, and instead of seeing red spots, we see green spots. So these are the green spots on the tummy of the macaque infected with measles. Now, this is the systemic nature of this disease. This is all of the gut-associated lymphoid tissue. These are the germinal centers in the spleen, and what you can see is this phenomenal infection of the lymphoid tissues, and you get this profound immune suppression, and what that allows is secondary infections to come along, and that's what causes the deaths, or predominantly those deaths of 8 million children were secondary bacterial infections. So the virus is very transmissible. The virus is also very, very acute and very, very immunosuppressive. This explains a little bit about the transmissibility of it. This is all around the buccal mucosa of the animal. At the later stages of the infection, you can see this profound infection where the animal is breathing large amounts of virus into the air. This is looking at the front of the buccal mucosa. Now what we've done is we sliced the head in two, and what you're seeing here is air coming in, and here, air coming out. So this is the nasal mucosa, the concha, the hard palate, soft palate, adenoid, and nasopharynx. And what you're seeing is how much virus is present in that upper respiratory tract and how much this pathogen is produced in these primates. And that's what we have to say about measles. The second thing which is important to remember is the virus is exquisitely a pathogen of primates. If there were enough monkeys in the world, it would be able to maintain endemic transmission in those monkeys as well. All right, so measles pathogenesis, acute, persistent. This is just a little cartoon. One of my mentors taught me so much. Bert Ream in Belfast and I put this together a number of years back, talking about the prodromal early acute established phases of measles. But what I want to just talk to you a little bit about is a part of measles that you don't really think about too often, and that is another thing I want you to remember about measles, which is the absolute paradigm for the long-term persistence of an RNA virus in people. And that is known in this condition called SSPE, subacute sclerosing panencephalitis. So this is another of my mentors from Belfast, a lady who was a tremendous individual in neuropathology, Dame Ingrid Allen. She passed away a number of years back. This is the paper she produced in 1967. This is some of her immunofluorescence way back then. And she taught me an awful lot about thinking about the brain and thinking about this condition. So this is some of the images we collected with Ingrid over the years. And you can see that the virus, whenever it reactivates in the brain after many years, is a neurotropic virus which predominantly infects neurons. Also infects astrocytes and oligodendrocytes, but predominantly it's a neuronal neurotropic virus. So we also then were trying to model this. We looked at it in animals. We used these fluorescent viruses. It allowed us to see where the virus was in neurons, allowed us to think about how the virus was getting around the brain. And it allowed us really to get to the stage where we were able to show and think about looking for longer term persistence. So this is some stuff which is unpublished, where we've not just made green viruses, but we made red and blue viruses. This is what they look like. One of the questions I've been interested in is, can exactly the same virus infect the same animal by different routes in different ways? Is there any cooperativity in the pathogenic process? And what you can see pretty clearly here is there is a lot of cooperativity whenever the virus is administered to these macaques intranasally, intraocularly, and intratracheally. Every single one of these syncytia came from an individually infected PBMC. So, for example, this PBMC, which produced this syncytium in vero cells, was from a cell which was infected just with one of the red viruses. This one here was infected with red and blue. So you've got two viruses cooperating in the same cell at the same time in the same animal. And then this one here, which is easier to see whenever I parse the confocal microscopy together, you can see that this particular syncytium, whenever we look at the individual images, has green and red and blue, and that's where we get the white here. So the virus can go in, the virus can cooperate, but what does the virus do in the longer term? Well, I've told you already that the virus is infecting the lymph nodes. So what we worked out in these animals was a way to do lymph node biopsies, pre-infection, 9, 13, 17 days, and then at necropsy day 22. And I wanted to look at that because there's been some suggestions that the virus is hanging around in the active form in these germinal centers. So RT-PCR, of course, tells you that the virus is present. Obviously, before the infection, it's not present. During the infection, it is present. But you can see at necropsy, some of the samples and some of the animals came up as positive. It tells you a little bit about the temporal presence of it, but it really doesn't tell you spatially where the virus is, and it doesn't tell you whether or not there's a very, very small amount of virus. So what we did is we developed RNA scope using positive stranded, negative stranded probes to allow us to look in these lymph nodes at the green, red, and blue viruses. And that allowed us to drill down, see where the viruses were, understand how they're interacting, understand that as well as being present in individual PBMCs, they're present in triply, doubly, and singly infected cells within the lymph nodes. And what we can do is we can look at how the virus is behaving, whether it be the genome, so that's the negative strand, and also the positive strand of the messenger RNAs in those lymph nodes. Then we can look at the whole lymph node at the time of necropsy. So this is 22 days after the infection. And what you can see here is this is the negative stranded probe. So whenever I drill down into this area and this area, what I hope you can see in these green cells here is very, very small numbers of cells still present within the lymph node. That doesn't prove or suggest that the virus is replicating. What we need to do is the equivalent experiment where we look at the positive stranded RNA. Same deal. This is a germinal center. One area of magnification, hopefully two areas of magnification. Hopefully what you can see here right in the middle of this germinal center is we still have a transcriptionally active infected cell in the germinal center at 22 days post-infection. And Diane Griffin, who passed away this year, actually is very interested in this in the longer term. She has evidence by qRT-PCR that the virus is persisting much longer. We have not as yet taken this out much further, but I think that's really important to understand what the virus is doing and what the virus might do, and why the virus might create lifelong immunity if we have these little localized regions where the virus is persisting for a very long period of time. All right, that's measles, that's negative, fast and furious. Now, positive. Pandemic preparedness, why we exist. Why we exist is whenever this coronavirus shadow comes around the world, people like us who are interested in respiratory viruses should be able to be active. We have a biocontainment lab. This is one of our colleagues. We were very fortunate, I talk about Christian already, to get the Munich virus directly from Christian in the early stages, Felix Drexler in the early stages of the outbreak. And we were able to do some virology. And again, in the very early stages, we talked about Andrew doing some of the similar things, we noticed all of these changes around the furin site. And we were like, well, what's going on? We don't know what's happening. We recognized that there were changes in both the Washington strain and the Munich strain. We have a lot of preclinical capabilities. We can do mice, hamsters, ferrets, 5 different sorts of monkeys. We chose the African green monkey model, and we set a PET-CT scanner in the facility that lets us look at the progression of the infection in the monkeys over time. And then this is, of course, at necropsy, just looking at where the virus is present. So we did the virology, we did the animal models. But what's really great about Pittsburgh is we have our university hospital is really interdigitated between CVR, my centre, and the people are just literally next door in ID. And this is an interesting story that I want to share. And I want to link it back to persistence for you. This was a long-term chronic right at the very beginning. So this is 2020, middle of 2020. Someone who had a CAR T cell infusion, got infected with SARS-2, admitted, was given convalescent plasma remdesivir, discharged whenever the QRT-PCR dropped sufficiently, went home, but was readmitted with this chest x-ray. And you can see the consolidation is significant here. He was intubated. He was given more plasma, convalescent plasma. He was given dexameth a number of times between day 63 and 71, and unfortunately he died. And at the time of his passing, you can see the viral loads began to increase. Now, we became aware of this after this detailed progression. After how we became aware of this first was this photomicrograph. The 2 clinicians walked in to my office one day and said, we've got this patient who, and I've explained what the patient is. This is an apical cell. Here's the cilia, here's the apical surface, here's the nucleus. And we see lots of this virus present in these endotracheal aspirates, which we've isolated, which we've obtained in day 22. And the question that they basically asked is, do you think you could isolate the virus from this? And it's like, well, heck yes, look at the amount of virus here in this. Sham is here with me. Sham is magical at doing isolations from the most complicated samples. This was really straightforward for him, and this is the virus that he grew quickly from that respiratory sample. We sequenced it, and this is what we saw right back at the beginning, right? Before people were talking about deletions, and we called them recurrent deletions, we saw these triplet deletions in the Pittsburgh patient. These are just 2 different samples from that person. And when we went into the databases, we started to recognize that there were multiple triplet deletions and multiple samples, and nobody was talking about this right across the world. And we called them recurrent deletion regions, and we identified them in this population of individuals. As I say, you had that lovely talk yesterday, which really winds on what we've done 3-4 years into the future. And they were all in this NTD. And yesterday you heard about the 6970 deletion here. That was what we called recurrent deletion region 1. We saw that they were predominantly present in viruses which were replicating. So these are all the in-frame deletions whenever we looked through all of these samples in GISAID. And we could see that there was evidence of transmission. And I don't have the time to show you those data. We looked at different areas of the world, and we saw that there was transmission occurring. Now, Sham isolated the virus. This is one of the first people that I employed as new faculty in my centre. He comes from Steve Harrison's lab, who's a structural biologist in Harvard and Boston. And he then was able to look at it and think, I wonder where these deletions are. We talked together. And of course, you see that the deletions were present in regions which were known monoclonal antibody binding. So the monoclonal which could bind was called 4A8. And it suggested that if this Pittsburgh patient had this deletion, and they did based on the sequencing, that they should no longer be able to bind RDR2 and RDR4. So we looked at that. We did the expression studies. We showed that if we take these deletions and we do the expressions, it still is bound by a serum monoclonal which reacts with the RBD. But whenever we use this 4A8, you can see that RDR2 and RDR1 no longer, whenever these mutations were made, these deletions were made, no longer bound that antibody, indicating that what we had seen is the beginnings of a true, actually one of the first antibody escape mutants in one of these long-term progressors of that disease. And this is just the data showing you that if we look at this with human convalescent serum as a positive control, you look at it with the 4-8A, we do the dilutions, this gets neutralized, the Munich gets neutralized really well, but the actual virus itself, whenever Sham did the PRNTs, the virus could no longer be neutralized, indicating that we had a bona fide escape mutant from a persistently infected individual. Now, I can't go into the whole details of the paper, but this is what we did, this is what we published, and this is what we were interested in showing, again, way back at the beginning of the outbreak. All right, so negative, positive. Now I'm going to show you both, and I'm going to show you a little bit of things into things. Everyone tried to bring their favorite idea to the table whenever we were developing COVID-19 vaccines. Of course, we know what was the winner. But this is what we did, and we published it just last year. And I just want to flag it as something which is probably the best way of looking at vectored measles vaccines, and why I actually think that because measles is such a wonderful vaccine, and because measles is such a great disease, that this approach, even though it seems so compelling to take a really good vaccine, puts things into it, and then use it to vaccinate people, just does not work, and actually probably doesn't have a lot of mileage. So we worked with these funders. We published the papers I'm going to bring you through. We basically took the entire spike and we put it into our measles backbones. And what we did first was we had to work out where was the best place to put it. Measles has an interesting way of expressing its genetic material. So there's more transcripts produced from the promoter proximal regions than the promoter distal regions. So we put it into what we call position 3, 1, 2, 3, third gene, or position 6, 1, 2, 3, 4, 5, 6 gene. And what was interesting whenever we grew that is you could see that the virus with it in position 3 was really growth restricted compared to the virus with it in position 6. So that told us that we should use position 6 viruses. Then, and again, we don't have time to go through all of this, we took all the spike and iterations of the spikes, the 2P mutations in the furin cleavage site, deletions in the furin cleavage site, the 2P mutations, deletions in the furin cleavage site, and then also mutating an endoplasmic retrieval signal, which is at the end of the genome. And we asked which ones of these were most immunogenic in mice. Now, remember, I've told you that the virus is exquisitively a pathogen of primates, so it really only infects primates, but we can do immunogenicity assays in mice. And for the sake of, again, time, I just want to show you that we homed in on this 2P prefusion delta furin cleavage site, and then the 2 alanine mutations, and that was the one that gave us the best immunogenicity in the murine model. Because the virus doesn't replicate in mice, and because we had this need to not go immediately into monkeys, we used this model, which basically allows a prime, a boost, and then we take ACE2 and allow the virus to be challenged, the ACE2 adenovirus to express the receptor, and that allowed the animals to be challenged. And then we looked at them 1, 2, 3, 4 days after challenge. And that experiment allowed us again to show that the infectious titer for all of the different recombinant measles viruses were reduced. But if you looked at the actual genomic RNA, both the delta furin and the prefusion delta furin 2A were equivalently efficacious. So we again chose this as our lead candidate to go into non-human primates. So non-human primate experiment, again, it's a prime boost experiment. We used the virus that I've told you about already. So this is what it is here. It's now in red for you. The control is in black. And we were able to use that non-human primate model, that African green model, to look at how the virus replicated and whether or not the animals were protected. The immunogenicity data was very convincing. The PRNT data, again, very convincing whenever we looked at these. And it was really clear that the recombinant measles was introducing or inducing a really significant anti-SARS immune response in these animals. Now remember, these animals are naive. They are equivalent to babies who have not as yet met measles virus, and that's the fundamental difference between this model and real life. We'll talk about real life later on. Replication kinetics were good. You can see that it was reduced oral, nasal, and rectal swabs. Again, there's a significant difference whenever we're looking at the vaccinated animals compared to the control animals. You can see in this image much more red here. And of course, we were able to use our PET-CT. We were able to look in the deep lung, and we were able to see that signal was really only present in the control animals, and actually in the early stages and the late stages of infection. We really didn't see replication of the SARS-2 in those lungs at all. This is just a little bit of immunohistochemistry. Again, nice, open, unconsolidated lungs in the vaccinated animals compared to what you saw in the control animals, and that's what you see here. So, mice said it was good, monkey said it was good. 2 slides on the study that we did, a randomized control study with Institut Pasteur, placebo-controlled phase 1 study for safety and immunogenicity. Again, you want to read it, here's where it is in the literature, but what I wanted just to specifically show you is that even though we had all of this preliminary data which suggested that it was really good, as soon as you put it into people who had pre-existing anti-measles virus immunity, they did not have a significant impact on the immune responses, i.e. the virus, even if they had been infected as a child or vaccinated as a child or vaccinated as an adult, the measles antibodies response was enough still to neutralize that vectored measles SARS and not give us a strong response. So these are all of the things that have gone into things in measles. Look at the list. It's long over the last 20 years since we started doing these things. And what I want you to remember from this is the measles vectored vaccine platform has consistently delivered for liver. Why is that? Because measles is a wonderful vaccine against the most infectious human pathogen on the planet. Okay? We're getting the theme here. All right. So we've done negative. We've done positive. This is Jules' fault, not mine. We've done both. Now I'm going to bring you back to negative. I want to give you a little bit of an idea of what we're doing at the minute. Again, this is all unpublished stuff. It has to be pandemic preparedness, not pandemic panic. And what we have been interested in, and this is NIH came to Becky Dutch and I, this is us just recently at the Gordon Conference in Barcelona. Becky and I were asked to bring together a group of individuals who would recommend a prototype pathogen for the paramyxoviruses. And why did they want to do that? Well, again, back to the people in this room, the people who have thought about MERS for a long period of time. The work that Barney Graham, Jason McClellan did with the stabilization and understanding the pre-fusion mutations in MERS, SARS-1, that was able to be used as a prototype and pivoted into the SARS-2 that gave us the RNA vaccines that worked so well. NIH wanted to take that approach for viral families which were not worked with as much, not flu, not coronaviruses, but flaviviruses, bunyaviruses, picornaviruses, togaviruses, and basically say, if you were to choose a prototype pathogen that would allow us to prepare for the next pandemic, as a group of paramyxovirus experts, what would you choose? So Becky and I pulled the folks together. We've published our recommendations, and the one that we decided that we should focus on would be the rubeolavirus mumps, because it's got a high R0, and the respirovirus PIV3 and 1. Again, human pathogen, respiratory transmissible, and emerging rubeolavirus and emerging respirovirus would give us problems. So therefore, we should be prepared. So one of the things that we know about the 2 components of the MMR, measles and mumps, in the vaccine, these are two paramyxoviruses. One of the things we're absolutely certain to be able to state is they're great vaccines, but they're slow vaccines. So the idea that we only have the ability to protect against a paramyxovirus using a live attenuated vaccine is too slow for pandemic preparedness. So we focused then, of course, on a collaboration that we have with Moderna. RNA vaccines have really changed the pandemic. RNA vaccines have so much potential to be used quickly. They might not as yet be as good as the live attenuated vaccines, but they certainly can be generated fast. So what we did was we looked at the mumps genome and we asked a very simple question. If we make RNAs from the current vaccine, remember this is the vaccine which was isolated in the 60s, we're still using it today, and we make RNAs based on the currently circulating strains of the virus, will there be a difference? And then the other questions that we asked were, if we try to put the fusion protein into the pre-stabilized form, right, copy what happened with SARS-2, will that help in making a vaccine, an mRNA vaccine for a paramyxovirus? And the difference between the paramyxoviruses and SARS-2 is the spike contains all of the components, receptor binding and cell entry for SARS-2, whereas we have a fusion protein, F, and a hemagglutinin neuraminidase protein in mumps, which binds the sialic acid receptor. So we took the RNAs, we made the RNAs, Moderna put them into their LNPs, and this is just showing you what the HN signal looks like, the F signal looks like. Whenever we take the vaccine RNAs or the wild-type RNAs, they produce these beautiful big syncytia within the transfected cells. Then we were hoping that by deleting the cytoplasmic tail or making the pre-fusion stabilized, cysteine stabilized form, we would ablate fusion. And what you can see is we really did that very well with this CC mutation, and even the delta tail reduces the amount of fusion that we get. So we had predicted in our antigen design that this would work. What we know from this is that the glycoprotein function has been modulated in the mRNAs. So the next question is, well, what about immunogenicity? So we did the same experiment. Again, we have the vaccine mRNAs and we have the wild-type mRNAs. We have a BALB/c model. We take the mRNAs, we put them in individually together in the lipid nanoparticles in a prime boost. We bleed them, sham bleeds them every at these time points, and then we determine what sort of responses we get whenever we administer these mRNAs. So this is a PRNT test. This is looking at the immunogenicity, PRNT 80 here, so we're going up to 1500. And what you can see for the vaccine mRNA is that we get a signal with the HN, which is superior to the F alone. So these are all of the individual molecules alone. And that's true for the vaccine mRNAs and the wild-type mRNAs. But what was cool was whenever we co-administered them, remember, these are not in the same LNP. They're put in LNPs which are separate. We get this much more robust response with the two molecules. And of course, that's obviously much better with the wild-type mRNAs. So dual delivery of the messenger RNAs generates a much more robust anti-mumps response. That was at 20 days post-vaccination. Now I'm going to show you 35 days post-vaccination. And the scale has changed. So the 500 that you were working with the last time is down here. Now we're at 25,000 in terms of the PRNT 80s. And what you can see here again is whenever we look at the wild-type virus compared to the vaccine virus, we get this enormously better boost and this greater neutralization whenever we co-express the F and HN. And there doesn't seem to be a phenomenal difference whether or not we have the delta CT or the stabilized fusion molecules. The big difference is between vaccine and wild type and between single and dual. So we can make RNAs, we can modulate the function of the mRNAs, but what about the protection? Do they actually protect against the infection? So the problem with mumps is also pretty much a pathogen of primates. It's been really difficult to generate an animal model for the disease. But what we did at the same time as answering this question is develop a brand new animal model for mumps, and that involves using cotton rats. So what Sham can do is infect these little dudes. He can isolate the lungs, he can inflate the lungs, and then he produces these lovely lung slices that allow us to look right through the lung and see where the virus is replicating. So this is a fluorescent virus that we use, so therefore you can use the fluorescent virus and you can see where it is. That allows us to really understand where the virus is over time. We can do nice pathology in it. So this is the virus replicating in the nasal septum, the neuroepithelium here. So it's really quite a nice little model. It's not a systemic infection like what I've shown you in the monkeys, but it's a good infection of the upper and lower respiratory tract. And we can pull bile cells out. And if the bile cells are infected, we know that the virus has really spread quite efficiently. All right, so got the RNAs, we've got the animal model. What about then the intranasal infection? So here's the cotton rats. Here's the wild type mumps that we're going to use to infect them. We prime, boost, and then we can do this 5-day challenge by intranasally infecting them and then sacking them at 40 days after the immunization. So now, this is complicated, so I'm going to bring you through it slowly. I'm going to show you day immunogenicity from the serum on day 20, 34, and 40. And I'm going to show you an HN ELISA. So this is antibodies against HN. These are the antibodies against F in this ELISA. And these are the controls, PBS and the randomized mRNA. And you can see that there's no signal here. So what you can see is at 20 days post-infection, we have a really strong signal for HN against HN, of course, and F and the FCC. We've not looked at the delta, the deletion. We're concentrating on this cysteine motif, stabilized F here. So you can see a nice reactivity here. And then you can see by day 34, the reactivity increases. And you can see by day 40, the reactivity in the ELISA increases further. Now I'm bringing in, and I'll flick backwards and forwards between these. So now I'm bringing in H and N and F by itself on day 20. You can see that we're already superior to H by itself here, and we're already superior to F by itself here whenever we're co-expressing these. Now this is day 34, and you can see that we're really now better than any of the individual molecules whenever we co-express them HN and F or HN on the stabilized one by day 34. And again, by necropsy time, we're really peaking. So what you have seen there is a robust immune response in the animals. But the question is, they make antibodies, are they protected? So here's the PRNT values. Before we go to that, this is PRNT 90. Whenever we just look at 20 days post-infection, this time we're only administering wild type. We've gone away from the vaccine mRNAs because there's really not a lot of point doing those. You can see that they just weren't as good. So here it is by itself. Here it is by 35 days post-infection. And then here it is in the cotton rats at time of necropsy. So you can see, again, co-administration of these wild-type mRNAs, we get this really good, robust response. So it's immunogenic, it neutralizes, but does it protect? And these are the data. I just use one of these images to show you that whenever we do the bile isolation, so basically isolate the bile cells, dilute the bile cells 1 and 2 the whole way down to 1 in 512. And you can see that in all of the animals, not a single infected bile cell was isolated whenever they were immunized with HN and F mRNAs. Whereas in the controls, PBS control and NT-fix control, we get a lot of virus out in that model. So we really have complete protection with RNA administration of that virus, in the cotton rats. All right. Last thing about the prototype pathogen is it has to go beyond the known human rubella And this is a really, this is one slide I'm going to show you. This is where we're starting. We're starting with a virus which has been isolated, which is rather similar to mumps, but it's been isolated from a bat. So what we've done is we've taken the fusion and the hemagglutinin and neuraminidase genes, we've made expression constructs, we've shown that whenever you express mumps, we get syncytia. Whenever you express bat mumps, we get syncytia. And then whenever we get the heterologous expression, so F from mumps, HN from the bat, we get small levels of fusion, but you can see that the F of the bat virus and the HN of the human virus really can complement each other very nicely. And again, this is linking you back to that thinking with these three colored viruses infecting at the same time, viral cooperativity in vivo, thinking about how new variants may arise. So what we have to do is we have to look at common themes with the prototype mumps and then ask the question, can we take what we learned from mumps and do the same thing for bat mumps? And then another rubella which is more phylogenetically, taxonomically distant and further and further. And where that is very helpful is to have my good friend collaborator works in, you've heard that already, works in Charité with Christian, Felix Drexler. Felix really published a seminal paper in Nature Communications in 2012, where he identified all these interesting novel bat paramyxoviruses. And actually there was one which was a really bat morbillivirus, which we've been working on. I don't have the time to tell you about that. That was the paper that he just published. This is one that we just published a couple of, actually probably just 4 weeks ago, actually, looking at neotropical bats and looking at the evolutionary trajectories of these morbilliviruses, these paramyxoviruses in these neotropical bats. So Desmodus rotundus is the vampire bat. So we have vampire bat morbillivirus. But these are the other two species here. This is gorgeous little thing here. I wish I could remember the name. I think yeah. This is the technical name for him, but it's some dog, riffing on some dog because of what he looks like. But we viruses, morbilliviruses, which are circulating in these two species, and that's all across South America. So what did we talk about? We talked about measles, the most infectious human pathogen on the earth. We talked about how our thinking as respiratory RNA virologists allowed us to pivot during the pandemic so that we were able to take advantage of what we did to really do useful things. And I really, I'm super proud of having looked at those, found those deletions very early on in the pandemic, because you can see that that's bearing fruit even to this day. We talked a lot about something. I think it's also good to show you things that don't work, right? Young people should see things that don't work or when to stop. This is when should things into things stop? Probably quite soon, right? So I'm not ashamed to show you things that don't work, because I want to show you things that does work, and new ways of thinking about old problems, old vaccines, like the live attenuated vaccines, as we've gone back to the negative side of the world and talked about pandemic preparedness. So Jules asked me to give this talk, and it was hard. This is our negative strand virology meeting. And what we did in the negative stranded virology meeting 2-3 years ago was we made a reverse complement program, because all of our negative virology colleagues had been working on SARS-2, and they didn't have a lot of negative stranded data to show, but they wanted to get together at the end of the pandemic. And my very good friends, Sean Whelan, Wendy Barkley, you know Wendy well, and I organize this meeting every 3 years. So we organized SARS Sunday before we had the negative stranded meeting. And why do I show you that? Well, I show you that because of this. This is from the booklet. And it loops me back to the beginning of the conversation, because it allows me not to acknowledge just my folks in the lab, but it allows me to acknowledge what you did for us. You supported, you helped those young early career researchers who had been pulled into SARS work but wanted to come back to negative stranded work. You allowed them to go to that meeting, you supported them with grants, and we had this in our booklet, because truthfully, if we hadn't had this, we wouldn't have had a positive meeting. We wouldn't have had a positive negative meeting without your support for this. I really wanted to flag that very clearly and thank each and every one of the management team who talked about that and gave us that grant. And then we're nearly finished. We are finished. Here's the acknowledgements. I talked about these people as we've gone through, but Sham's in the room. He doesn't often get to see me present at this part of in this way. So if you're really interested in any animal models, he's a wonderful array of animal models that he's doing, and he's really interested in people, MERS collaborations in particular, as we work with SEPI. And again, the rest of my group in the CVR, and actually many of my faculty in CVR, have been instrumental. And that is me.
Thank you, Paul. That was a really wonderful, wonderful talk. Questions are on. Over there at the back.
We should get them to self-identify as negative or positive before they ask the question, right?
Both and
or both.
Super as always.
Thank you, Darren.
I just wondered about the use of measles as a vaccine vector. I mean, obviously you were saying it doesn't look too good, but I wonder about measles as an oncolytic, whether or not you feel the same obstacles are there for its use as an oncolytic, because I mean, I've seen some positive early results from that, but I don't know if you're more aware of where we are in that field.
So what's really interesting about measles as an oncolytic is one of the challenges with these viruses, whenever you start mucking around with them, they really don't want to have big proteins in them or express big messenger RNAs. And you saw that with the position 3 and position 6 virus, right, with SARS-2. It just didn't want to grow very well. So those oncolytics don't grow well, deliver big cargoes, and the only way to use them is to really produce, it's something like 10 to the 9 or 10 to the 10 that they inject into people. So remember, you're doing something very different, right? There's a fundamental difference between an intramuscular immunization with measles and an intratumor installation of 10 to the 10 recombinant measles. So I don't think that the immune system has the same ability to knock 10 to the 10 on its head as it does to have a normal dose of measles vaccine. Even if we, because remember, a normal dose of measles vaccine is only 10 to the 4. That's not a lot of virus. And the only reason it works so well is because it's a live attenuated virus. And the only reason why the oncolytics work so well is because we literally swamp the tumors with them. Oh, sorry.
I saw Julian's hand first.
No, we don't want questions from him.
Julian and David.
Well, that's the one thing that we can all say in this room, right? We're never going to go onto the dark side. No. Actually, Volker unfortunately has gone, and I can say it here, and then whenever somebody steals the title, you'll know it was my idea, right? Volker and I have committed at some stage, whenever we've nothing to do, which you can imagine that's not going to be too soon, I want to write a paper, a review called "Are Live Attenuated Vaccines Dead?" Right? I want to. So you've heard the title from me. If anyone steals it. It's a trademark type. I actually don't think so, Jules. In fact, what we know is, whenever you get to this stage of your thinking, I see the live attenuated vaccines as being so good, and the mRNA vaccines are not. And what I would really love to do in a sort of a systems biology, omics-based approach is try to bridge the gap, try to work out what the live attenuated vaccines can tell us, how they can be used to teach the mRNA vaccines to be better. Will they ever be as robust? Will they ever be as good as measles? The day and hour we make an mRNA, H and F for measles, which gives us equivalent to the live attenuated vaccine, the answer to your question is yeah, they're done. But I believe that's a long ways away, which is why I think riding both the horses, mRNAs are fast, reliable, safe, and the 100-day mission could be fulfilled with an mRNA. I actually think it'll be interesting to see whether or not the protein folks are able to do it too. They might give the mRNAs a run for their money, but absolutely no live attenuated virus is going to be able to be ready, produced, tested, and ready to go in 100 days for SEPI. So the answer is absolutely both, Jules.
All right, I think in the interest of time, we'll move on. But that was fantastic. Thank you ever so much.
Thank you.